Hi, in this video I'm going to be reviewing week 2 activity 3. So here we're in week 2, and if you go into weekly academic content, alright, we're going to have weekly activities with Professor Johnson, and then in there you're going to find activity 3. This video actually will go into this folder the video we're making right this minute. But what we want here is worksheet for activity 3. All right, I'm going to open this file and I'm going to review this with you. You will need to complete this one and turn this one in as an assignment. So whereas the other one, I just kind of asked you some simple questions to keep it easy the first week, this week you're going to actually have to complete this and upload the answers in the under the assignments folder. All right, and I'll show you that at the end of this video. So first, here we have nucleic acid directionality and polymerases. Essentially what we're going to do here is more or less define a gene, though really the concept of a gene has changed over the years. So this particular, the way I'm describing it here is not necessarily the way that a more advanced molecular biology course or evolutionary biology course might define a gene. A gene might just be any region that has some functionality. Here we're going to stick to a pretty simple, straightforward one that's kind of stuck in everybody's head because of the central dogma, which is also not in favor these days. But we're going to say a gene is simply the coding region flanked by a promoter and a terminator. And we'll talk a little bit about what those pieces are. We've talked a little bit about promoters here in different sections for this particular week. But in general, before we do that, we have to talk about directionality and realize that the DNA has chemical directionality by these structures on either end and how how the bonds are located between the various nucleotides. As you can see here, we designate one end the 3' end and the other end the 5' end. That would be for this particular single-stranded DNA molecule. For a double-stranded DNA molecule, the complement, as you know, C binds to G, T to A, etc. But also know that in one of these, it's going to have a 3' end and 5', and the complement will have the opposite prime number. If this is 5, this is going to be 3. If this is 5, this one's going to be 3, etc. So keep that in mind. Directionality is important because the polymerase, which does transcription, is going to move along these strands in a particular direction. Here, that's what we're getting to. Let's see down here. Nope. Let's see where. Right here. Notice that this top strand, 5, the complement, that's what I just said. But the polymerase, all right, moves in the 3 to 5 direction. That's the direction that it reads it, and it creates the new DNA 5' to 3'. Now, this is often very confusing in textbooks. They will say it one way in one textbook and in a different way in another textbook. The idea is instead of saying transcription occurs this way or that way, I prefer to say that it reads one direction and writes in the other one. So we will say that polymerase reads the original strand of DNA 3 to 5, and it creates new DNA from 5 to 3. All right, so that's the way I say it. I think that's clearer than the way you often hear it in a textbook. In this particular exercise, you're just going to do as we did earlier. You're going to find the complement here. Very easy. Nobody should have a problem with that. But you do need to designate the correct 3' and 5' ends. You need to understand what causes the polymerase to stick. What is a promoter? The simplest promoter is the TATA box, TATA, and a terminator, which consists of a hairpin loop. You'll need to read here about what a hairpin loop is. That will be very important for our exercise for annotating the lac operon. That's another assignment for this week. You should do this one first, just to get an idea of what we're calling a gene here. You need to understand that TSS, if you see this abbreviation, that's the transcription start site. That's not the same thing as the promoter. Now, the transcription start site might be enclosed within a promoter, and a promoter is a region of DNA, and something like a TATA box, which we often call a promoter, is really just a small piece of it. You might call this the polymerase binding site. There are lots of things you might call the TATA box or its equivalent. It's generally taken on the name of a promoter, but now there are many parts of a promoter that we know of. There's the core promoter, there's the extended promoter. In bacteria, it's a lot easier to just call the TATA box a promoter, and so I'll use that convention as long as we're talking about bacteria or prokaryotes. But when we get to eukaryotes, it gets a lot more complicated, and I'll have to use more technical terms when we're talking about the regions that polymerase binds to. Now, the region that it binds to designates it essentially as a gene. So the polymerase binds here, reads the DNA here, it makes the RNA as it's reading it, and then when it gets to this terminating region, it falls off and it terminates and stops making the RNA. So the polymerase falls off, the RNA falls off, you're done. Now, what causes it to fall off? A hairpin loop. This is a hairpin loop. It's a piece of RNA here in this particular case, the single-stranded RNA that binds to a nearby region. This nearby region will be separated by a piece that it is not complementary to. So in other words, you have two complementary areas that near one another that can bind to one another. You can think of this sequence more like a sheet of like a sheet on a line that's blowing in the wind, or maybe a flag or something. The RNA here is not straight, and when these two regions get near one another, because they are flexible, they essentially bind to one another and form this loop. All right, with this region being the loop at the top. This is the binding. This is the stem part, if you want to call it that. And this is a hairpin loop, and this forms in the RNA, not in the DNA, and it causes the transcription to cease. It causes the polymerase essentially to fall off. So that's what you would look for here in the terminator region. The promoter region, at least in a bacteria, you're primarily looking at TATA boxes or something similar. You need to understand these things. You need to understand that these essentially are pretty much the same in eukaryotes. We have a lot more to offer in the structures for the promoters. There are many, many, many promoters, and the region is much longer in a eukaryote with a lot more regulation. The termination sequence or the termination signal, it can be a little different, but you still have primarily hairpin loops or some other structural change in the DNA that causes it to fall off. There are sequences that bind proteins that cause the polymerase to fall off in eukaryotes, and at least Rho in bacteria also enhance the terminator hairpin loop. We'll talk about that at another point. So here's our gene. If you are talking about eukaryotes, that sequence we just looked at is going to be here on the chromosome. Here we're just talking about the fact that eukaryotes, of course, the RNA is created and it leaves the nucleus to go out and more or less find a ribosome, which will bind to the 5' end of the RNA. That is not what happens in bacteria. In bacteria, the RNA is created and stays on the bacterial chromosome and kind of attached, and ribosomes come and find that RNA, and that is different than what occurs here. If you don't understand what I'm saying, you need to look up the differences in transcription between eukaryotes and prokaryotes. That is important to understand. Here we're just finishing up this particular worksheet. I'm asking about how mitochondria and chloroplasts, how their genes function differently. They function actually very similar to bacteria. And if you're not familiar with the endosymbiotic hypothesis, you ought to look that up and to answer this question number 5. And that is